Hi everyone. Since this week's lab is talking about how to read an article efficiently anyway, and since all of you are going to have to do a primary article presentation in this class at one point or another, with your teams in the groups as part of the units, I figured I would also give you some tips and an example of a good presentation. So when I think of presenting an article, I really think about how the article itself is formatted. So I think in terms of an hourglass. So when an hourglass starts out wide, it's very broad and then kind of gets very narrow and very specific, and then it's going to broaden out again. So this is often the way a lab report, a article, and a good presentation should be. You're going to start out broad, so you're going to put the article into full context. What's the background information that's important? What's the question they're answering? Why are they doing this research? All right, and what information outside previous information led to this? And so if you're starting really broad, what's the context? What's the background information? What previous information was there? And then by the end of that, you really have the key terms, the key information that's really going to be needed to understand the article and put it into the context of its importance in the broader scope of science. And so when you're doing this, you need to really understand the background information. You might even need to find some outside information and bring that in. So if there's some terms you don't understand, bring in some outside information. If there's some previous work that was really important to understanding this article, go find that previous article and at least maybe bring one or two key bits of data that led to this and so forth. So really think about what's needed for someone to really understand this article and its context and its importance. Okay. And then from there, obviously we have methods. So this is where you really don't have to present methods in great detail. So for example, if there's results that used a Western blot, you don't have to go through 5 minutes describing how a Western blot is performed, things like that. You really only need to say something like a Western blot can be used to determine the protein levels in a cell. And so here they're using a Western blot to compare the levels of protein A before and after this treatment, whatever it might be. So as long as the audience knows why that technique was used and what the data is showing based on that technique, you're going to be fine. And so I usually don't spend a lot of time going over a bunch of methods early on. I just kind of really very briefly introduce the method right in front of the experimental data, the results that kind of used. The exception to this is maybe there's a really key method that they use throughout. So if there's a really key method, there's a method that you're going to see over and over and over again, then you might want to spend a little more time up front kind of just describing that method so that with every figure you're not saying the same thing over and over again. And the other exception is maybe if it's a very unique method. So if it's not something more standard like a Western blot siRNA, but they really had to devise a model system for the experiment, that's where you really might want to put a little bit of time in describing what the model system they developed was. Because if they developed a model system and this is that unique, that's actually a part of the article in itself. All right, so then the data, the results. This is very important. You really want to walk the audience through the data. And what I mean by this is when you're describing the data, actually point it out, right? If you're describing a graph, make sure you are actually pointing to the control data and then comparing that and pointing to the test data and so forth. You also really need to make sure that at the beginning of that results section, you're very much telling the audience what the authors were trying to determine. So what was the question that this figure is answering, right? And so within that then allows the data to fit into that question. So here's what they were trying to show, and this data shows that, boom. And so I already kind of talked about just very briefly introducing the method, or at least what the method allows them to see. And then the key aspect is the take-home data. So after showing all those results, what is the overall message that they were showing? What's the take-home message? What's the key conclusion from that data? And then this is going to help you transition from one to the next. So if you've just said the take-home message from figure 1, now you can start with the question for figure 2. So now that they've determined this take-home message of figure 1, they then wanted to know this question of figure 2. Describe figure 2, show how it fits with that question, and so forth. And so in conclusion, figure 2 overall shows us this. Now that they know this, they wanted to show this. That's the question for figure 3, and so forth. And so if you keep that going, you're going to really guide the audience through that information. And then finally, discussion. You really want to summarize key findings and relate them back to the big picture. So 2 things that I think are really important. People do a very good job of summarizing the key findings. They often have bullets and so forth, but make sure you really, again, put those into context with what they mean and why this was important in the first place. And this is really key. Oftentimes, there's a set of things they've shown. Otherwise, the whole article would be just one figure. But figure 1 kind of showed this, which then figure 2 showed this, which added on to it, and figure 3 and figure 4. And oftentimes that's even kind of a series of things that they learned. And so if you can create a schematic, if you can create a visual interpretation of kind of how all of those different figures fit together into the big picture, that is absolutely huge. And you'll see this sometimes in articles themselves, but I really think that schematics say a ton. Remember, a picture is worth 1,000 words. So if you have a slide that just has a whole bunch of bullets, like I do here, so this is probably not the best thing to do, but I want to give the overall main points. If you can kind of figure out how to put that into a picture where you're describing that along the way, that goes a long, long way. And then also don't forget the next steps. Science is never done. For every question you answer, there's several more that then become asked. Right? So they now know this. This is kind of, this transition is almost like from figure to figure, but it's for the whole article. Now they learned all of these things, but they still need to figure out this, or what's the next step, what they still want to know this, or we need to now figure out how this works. What are the next steps? And then finally, strengths and weaknesses. This is probably the hardest part for undergraduates, because I know when I was an undergrad, my thinking was, who am I to find any weaknesses in this article? You know, these people are scientists and I'm just an undergrad. But every article does have some weaknesses, right? This is part of science, and that doesn't mean the article is bad, but there's going to be some really strong aspects to it. Like, this is strong, that conclusion is very, very strong. And then there's going to be some where, okay, the data was pretty good, but maybe there was a slight weakness in the experiment, or, you know, the data doesn't look completely convincing, or more importantly, there might be alternative explanations. It's not that the article was bad at all, but it's still possible that another explanation for seeing these results might be this. So try to think through those. That's a big aspect of being scientists. Being a little bit skeptical, not mean skeptical, and not just kind of absent-mindedly thinking that everything's wrong, but are there other explanations? If I see that data, could it mean this instead? Or maybe not. And so see if you can find some of those and present those. All right, I'm going to stop this video here. These are the kind of overview things, and then the next video is going to be me actually demonstrating this on an article so that hopefully you can see what this looks like. Thank you.